Well, good afternoon and welcome. I'm delighted to have you here. We have a special treat today. Siddhartha Mukherjee is a researcher, clinician, and best-selling author. His book, Cancer, the Emperor of All Maladies, won the 2011 Pulitzer Prize for nonfiction. The New York Times described him as one of the most important writers and thinkers of our time. He grew up in India in a multi-generational household. His father was an executive with Mitsubishi, his mother a former teacher. His grandmother was a powerful influence. An uncle in the house was diagnosed with mental illness. From a very early age, he trained as a singer in the Indian classic tradition. He was drawn to science and medicine as he got older. Influenced by his grandmother's fatal illness and the cancer death of his favorite English teacher, the idea of writing a book came to him when he was training at the Dana-Farber. A colleague urged him to have a life outside of the hospital. Good advice. He wrote The Emperor of All Maladies over the course of 5 years. At the same time, he was starting a family and establishing a career. The New Yorker called it an extraordinary achievement. His second book is called The Laws of Medicine, Field Notes from an Uncertain Science, the subject of imperfection, uncertainty, and the future of medicine. His most recent book, The Gene, An Intimate History, another bestseller. Bill Gates put it on his list of recommended reading in 2016. His research parallels the subject of his books. His lab at Columbia University Medical Center studies the biology of malignant and premalignant diseases of the blood. His research was published in Nature, the New England Journal of Medicine, and other peer-reviewed journals. He was named among Time Magazine's 100 most interesting people in the world. He lives in New York City with his wife, a sculptor, Susan Z. They have 2 children. Sarah Z. Dr. Mukherjee enjoys cooking, continues to practice and perform Indian music. Last month, he began his new role as a medical columnist for the New York Times Sunday magazine. This video will show you more.
The knowledge of medicine, the power of words, the determination to discover a breakthrough. Siddhartha Mukherjee, an oncologist with a unique combination of inquisitive research and dynamic writing ability, advances investigation into understanding, all in the quest for a cure. Born in New Delhi, India, Siddhartha Mukherjee showcased his academic skill early in life. After achieving his school's highest award, the Sword of Honor, he went on to become a Rhodes Scholar, graduating from Stanford University, the University of Oxford, and from Harvard Medical School in the year 2000. After his residency in internal medicine at Massachusetts General, he trained in oncology as a fellow at the Dana-Farber Cancer Institute. Preparing the next generation of physicians, Dr. Mukherjee has been assistant professor of medicine at the Columbia University Medical Center since 2009. Filled with curiosity throughout his career, Dr. Mukherjee gravitated toward the lab. He investigated cellular genes that change the behaviors of cancer cells. Putting that research into words led to a breakthrough in Dr. Mukherjee's career. In order to find the future of medicine, he decided to look into the past and wrote his Pulitzer Prize-winning book, The Emperor of All Maladies, a biography of cancer.
When I met Sid and read the book, I knew this should be a public television documentary.
What Sid's books done is bring it to the fore and said, look, we must talk about it.
His writing and research took a personal turn with his latest work, The Gene, An Intimate History. Siddhartha Mukherjee explored his own family history to see what lies within our DNA. For his advances in medicine and science, the government of India conferred him its 4th highest civilian award, the Padma Shri, in 2014. Dr. Siddhartha Mukherjee takes a question and accepts the challenge. In an attempt to find a solution, he delves into the most intricate systems in the body. With dedication to investigation, his hope is to one day cure, control, and conquer cancer.
Please join me in welcoming Dr. Siddhartha Mukherjee. Well, needless to say, we're delighted to have you here, so thank you very much for joining us.
My pleasure.
Let me start with sort of a little bit of your background. Tell me, why did you decide to become a doctor, and when did you know?
So my decision to be a doctor, I sort of came in reverse in some ways. I trained as a scientist first. I was in England, and I was training in immunology. So I trained as an immunologist, and one of the projects that I was given as a young graduate student was on a virus that I knew nothing about called Epstein-Barr virus. Now I know quite a lot more about the virus, but as it turned out, I was looking at immune responses to viruses and became more and more interested in sort of where did the immune response begin? How does it begin? And so I actually went backwards from science. I'm saying backwards because many people in medicine go into the sciences after having seen human physiology and then enter the world of the basic sciences. I went backwards. So I became very interested in that. And of course, we've talked about a little bit before, I had a family history that was that pushed me towards thinking about medicine. We've talked a little bit about it, and those two things really came together for me in medicine.
But why cancer?
Well, again, the challenge of cancer was so great that it seemed as if it was the one thing that beckoned more than anything else. This was.
Well, you could have been a heart surgeon.
I could have been a heart surgeon. I tried. I tried. Cancer was my rejection choice. No. The fundamental problem of cancer, not just the scientific and the physiological problem of cancer, but also what went on, the larger, the swirl that we found ourselves in the 1990s when I was training around cancer, how to think about it, what to do about it. What is it? How is our conception of what cancer is? How is that changing? All of those questions were so seductive at that time. And most importantly, and I hadn't anticipated it, the way that your relationship with patients as an oncologist. Certainly you have that you have some of that as a surgeon as well, of course. But the idea that you would have this kind of intense, like a chemical bond, was very attractive to me.
We wrote two major tomes, and both of them you talk about them in terms of a biography of a disease or a gene. Why a biography?
Well, I think the word biography applies more than a history or some more anemic word, because the idea of illness is so close to us. I was particularly inspired by Susan Sontag's writing, for instance, where when you think about a history of an illness, when you think about the story of an illness, or in the case of the gene, the new book, when you think about that story, it comes closer, it cuts closer to who we are. And almost defines, in the case of cancer, defines to me a kind of alternative self. Sontag talks, and it's the quote that opens the book, she talks about illness as the night side of life. You inhabit the day side of life during the day, and the night side of life at night, illness being the night side of life. And I thought to myself, this inhabitation, day and night, suggested to me an alternate life. It was, and therefore the word biography. In fact, the name biography, I thought for a long time, and the name didn't occur to me for the first book, the subtitle especially, didn't occur to me until much later. And then when I hit upon the idea, the word biography made so much sense. that it came much later in the genesis of that book.
Well, a lot of us know the history of Mendel and Darwin, etc. But, I've always been interested in where the word gene and genome come from.
Right. So it's an interesting story. Mendel didn't use the word gene. In fact, Mendel had no idea, really, of the existence of what we would now call genes, even though he saw it in this abstraction. What's astonishing to me about looking at that history is how much even people who assume the words, even people who think they have a kind of fluidity with the concept, how much we don't know or didn't know about the origins. And of course, that tells us how much we don't know or didn't know about the future. The Mendel's genius, at least to me, was that he fused a particular kind of mathematics with biology. People had not thought about biology in this mathematical way because the themes, the fact that there would be interior harmonies across inheritance was really absent. So when Mendel read his paper, it's an interesting story, when Mendel read his paper in the 1860s, he read it out not to some very august body of the Royal Society of Science somewhere. He read it out to a group of sort of agricultural, basically a group of farmers interested in breeding. And his topic, what he had brought to heredity was what you would call big data today. He brought big data to heredity. He basically shelled tens of thousands of peas and brought that idea. He fused a kind of mathematical, statistical way of thinking, and of thinking about biology, and no one had done that simple thing before.
You did a lot of sort of basic research. You went to visit where he was.
Yes.
And where he'd done his work.
Yep.
And you had a little difficulty getting in there.
That's right. So, there was a small. I wrote a small piece about going to Mendel's monastery. What's interesting about it is that, again, we live in an era in disease where our thinking is dominated by genes and genomics. But no, very few people, maybe you can show of hands, very few people have gone to the place where all this was discovered. So my question was, why don't I just go there? Why don't I go to Mendel's monastery? And I was going to spend a week there. So I went to Mendel's monastery, tried to spend a week there, and the woman, the woman who was at the front desk said that I happened to have arrived they were going through some renovation, it was closed. And so I said, well, but you don't understand, I've come from so far away, there's no way I would have known. Can I not be let in? Is there any way? And she said, well, you have to apply in triplicate. You have to do, so I said, well, who do I apply to? And she said, to me. So, coming from India, I knew this ruse was known to me. So I said to her, I said, as formally as I could, I said, I hereby apply to you. And she was so stunned by the idea of me taking, I took a little bow, I said, I hereby apply to you. And then she let me in. So that was, I said, two could play at this game.
Speaking about playing at the game, the sort of the modern era of genomics really started in 2000. Mm-hmm. In one of your books, you make a wonderful quote, and I think it applies to some of the individuals involved there. You said that modesty is a virtue, but you get further without it. Do you want to describe that time and some of the people we know?
I refuse to take any names, but it's an important idea, I think, in human history. The draft sequence of the human genome and the things that have occurred since then in terms of our ability to influence, change, read, and write genes is an extraordinarily ambitious and also an extraordinarily arrogant idea. It lacks a certain, it has a hubris about it. We are becoming, in the words of John Silston, a machine that is learning to read and write its own instructions. what is life like for a machine that reads its own code and then begins to write its own manual? These are questions that are extremely important. They're circulating with great importance through the world of medicine. What if we were able to diagnose diseases before the diseases occurred? How much that would empower us, but on the other hand, what it would do to ourselves, our conceptions of ourselves. There's one word that sends a kind of chill up your spine, which I have a chapter called with that word. It's the word previvor. A previvor is a survivor of a disease you haven't yet had. But imagine your life being, on one hand, medicine being able to, imagine being able to run a trial before a disease occurred so that you could really understand intervene at the first step of the disease. But on the other hand, do we want to enter this kind of era of surveillance, of anxiety, of previvorship? These are questions that are extraordinarily important to who we are and what we're getting into in this new era of advanced reading and writing of the human genome.
But the magnitude of this discovery and this development is 3 billion base pairs.
Yep.
Give people some idea of the magnitude of that number.
Well, so one way to describe it is if it was written as, if it was printed as in standard font, a single person, your genome, would be 72 sets of the Encyclopedia Britannica, 72 full sets. So every wall in this room would be covered with one person's genome printed in kind of an encyclopedia format. It would actually stretch into a couple of other rooms as well. So that would be the library of yourself. You would essentially, you could go and pick out one volume of this 72 full sets of the encyclopedia, and it would be inscrutable to you. It would read ACTG, CCTG, GGCT, etc. That's not real. I just made that up. But the idea that from that code of codes that you can launch from a single cell this thing that becomes us. Of course, chance plays a role. Of course, the environment plays a role. But the fundamental determination of who we are arises as a consequence of that is to me very, very interesting. And of course, remember that the difference between you and a person who has a lethal disease, or potentially lethal disease, like Huntington's disease, could be, 1/2 of one page of that encyclopedia, of that full encyclopedia. The relevant difference between you. The relevant difference between you and a woman who has a 10-fold risk of developing breast cancer or ovarian cancer in her lifetime may be one word in that 72 sets of the Encyclopedia Britannica. And the capacity to find those words that make the difference, and then act on those words, is, I think, what we're learning how to do, and obviously has implications very widely for human beings.
Well, that brings up the topic of a huge amount of data and how are we going to keep track of it?
Right.
Which is can you imagine now that in cancers you're now sequencing both the individual and the tumor?
Yeah. And so, by the way, just to let you know, so I'm mostly a leukemia biologist, a leukemia geneticist. I work on blood cancers. Virtually all my patients that I see in clinic will undergo some sort of sequencing. All of them, 100%.
100% of them?
Yeah.
So how do you keep track of the data?
Well, so now, obviously computational power has helped. What's interesting is no longer how do we keep track of the data. That has become partly solvable. It's the question of how we make sense of the data. Those are totally different things. To make
Absolutely.
sense is the really key finding, or the key piece. I was very taken by a recent article, which has not even been peer-reviewed yet. These days, you can, as you know, you can put things out before they're peer-reviewed for public review, for wider peer review. So anyway, I'll talk about this for a second because it has many implications for disease. It'll take me just 2 minutes to talk about it, but I think it's very, very interesting and important. A computational geneticist interested in in height asked the question, can you predict height from the genome, knowing nothing else, just knowing the sequence of the encyclopedia, as it were, for an individual? Now, we know height is highly heritable. We know this because of 1,000 studies. Tall parents tend to produce tall children. Identical twins tend to be of similar height if you remove nutritional constraints and illness. So what this paper sets out to do is to ask the question, can you use the genome to predict height? Now, what's interesting about it is that until now, we knew several genes that were involved in height. But if you added the sum total of their contribution to human height, you'd only get very little. You could only explain very little of the variation in human height based on the known influence of the genes. So this team took an interesting approach. They said they went to a biobank, the UK biobank, and there were 500,000, maybe more, individual genomes and individual heights cross-listed against each other, all anonymous. And they said, what if we unleash deep learning on these 500,000 variations in the human genome? It's not full sequence, but variations and individual heights. And essentially, by attaching connection weights to each of the variations, they could get to within 0.5 centimeters of actual height. So in other words, you could essentially input a series of variations in the human genome, and you would output within 0.5 centimeters of actual height, the predicted height. And when they went back to ask the question, well, how many genetic loci or how many variations were involved, it turns out the number's not known yet, but it's on the orders of thousands, if not tens of thousands. Right, so this kind of experiment raises an incredibly important series of questions about disease and about how we think about ourselves. One question is, what if we began to predict this across other features? Where does one stop? What are the limits? What are the computational limits that allow us? How deep can we go? Can we predict other biometric features? And where do we begin to predict dangerous biometric features? Where do we stop? Who tells us when to stop? So that's one. And then from the standpoint of medicine, imagine if there were tens of thousands of variants that were predicting the risk of cardiovascular disease. Where would we put all this data? Would you carry it around with you so that you would know future predictions of disease? Again, going back to this idea of pre-vivorship. So the not just the data landscape, but the ethical landscape that's opened up with these kinds of experiments, combining deep learning with our understanding of genetic and genomic diversity, I think is going to be a series of questions that will really spend a lot of time in the next few years.
Are you familiar with Craig Venter's work on FACE?
Absolutely, yeah.
Why don't you tell him about that?
So, I should say that work, I read it because I'm interested in genetic variation from the standpoint of cancer. That work's been, there's been reviews and reviews of reviews. It's still in an arena where needs to be vastly more polished. But the hypothesis was that, again, could you use genetic variation to predict fundamental features of the human face? And the answer is yes, to some extent. We'll probably refine it. It seems to me that there are people who are critical of it right now because it's not absolute. But do I think that within the next 10 odd years, this kind of work will change, for instance, criminology? The answer is absolutely yes. I do think you will be able to predict maybe not the real face of the individual. That may sound a little far-fetched. But maybe you'll be able to predict fundamental features, for instance, their height. For instance, something about the general structure of their body. Something about the kind and texture and the color of their hair. Something about the texture or, something about their eyes. And then, subsequently, many, many features that open up ethical landscapes where we really, really need to think about this. Is it going to be a kind of, obviously this is genetic profiling. The word profiling is inescapable here. And remember, we're sitting in a hospital. Profiling is what we do in medicine. Whether to fundamentally to profile, to understand risk, is one of the goals of medicine. One of the goals of cancer is to understand risk. where does our understanding of risk then stop when it enters a social arena where risk becomes profiling? Those words are abutting words. Where do we draw those strong lines? These are, I think, important questions. I tried to answer them a little bit in the gene, but I think they're important questions, obviously.
Well, we're talking about profiling. Have you been sequenced?
I have not been sequenced. I have not been sequenced, partly because, just to let you know how trivial it would be for me to be sequenced. Our laboratory does sequencing on, I would say, daily basis. Exome sequencing is something that I do daily, sending out exome sequencing on a daily basis. So I have not been sequenced for several reasons. Most importantly, because of my personal history. I don't want to know. I don't want to know the risk alleles for mental illness in me. Computationally, I have a 10 to 20-fold increased risk for bipolar disease and schizophrenia. I don't want to know, but more importantly, I don't want to know the risk profiles of my children. I have 2 daughters, and if I knew, I would fundamentally begin. The question I ask myself is, what if only one of them had even a marginal risk profile? We know very little yet. We know that schizophrenia is going to turn out to be much like many other complex traits in which there'll be not one, but tens of thousands of what I call nudge effects, nudge genes, as opposed to shove genes.
I never heard that. That's a scientific expression.
Well, I could make it into a scientific expression, and then it would sound very, very, very complex and abstract, and no one would understand it. But that's basically what I'm talking about. One of the things we're finding out about human genetics is that many traits, not all of them, many traits are dominated by nudge effects as opposed to shove effects. That's very important for disease, but I don't want to know the nudge effects in my children, because what if one of them tested? It would fundamentally change the way I think about the 2 of them. And this is a question, of course, all of you are asking yourselves, right? What if one of your 2 children, yourself, your spouse, your parents.
How about your children? Would you have them sequenced?
No, I would not. I would not until, here's my, obviously there's a line between them. I don't want to know about nudge effects that have no medical consequences. If I can't do anything about it, I don't want to know about it. And this is actually, it sounds like the ostrich burying its head in the sand, but in fact, this is a very refined medical principle. The refined medical principle is, unless it's actionable, information, unless it's actionable, particularly as it pertains to individuals that we care about, is often information that we don't need to know now. For a research study, yes, but I'm not. My children are not a research study for me.
So how about pharmacogenomics? That could affect your kids.
Yes, it could affect. So again, just going back to the same example, the number of instances in pharmacogenomics, as you know very well, the number of instances where you have such a powerful effect that it would radically change your decision making right now are few and far between. There'll be more coming. Until that information becomes actionable, I'm not interested. When it becomes actionable, when I find out that that within the code of my children's genome there's an actionable variation on that will make a big difference to them medically, then I'll be inclined to find out.
Interestingly, I used to, when we used to anticoagulate people with warfarin, I would be there, and some people, you just wave a Coumadin by the bedside and they'd be anticoagulated. Right. Other times you'd be shoveling it in for days and no effect. And clearly that's genomics.
Right. What's interesting about that, for instance, yeah. So what's interesting about it, and this will be the case across multiple drugs. I mean, I'm extraordinarily familiar with this because this is very much the case with cancer. In some people, of course, you give them standard cancer therapy and they have very little impact. Other people, the tumor melts away and you have a powerful response. How wonderful it would be to be able to predict from a patient's tumor and they're normal, sample the capacity to predict how they would react to medicines. That would be a wonderful thing. But until it became actionable, I'm going to resist. When it becomes actionable, I'm not going to resist. I'm a geneticist. I'm a cancer geneticist. This is what I live by. But right now, especially for mental illness, which is the one thing that is, of course, the most relevant to my family, I would like to resist.
So let's talk a little bit about CRISPR.
Uh-huh.
And you can probably, some of the people in the audience may not know what CRISPR is, but maybe you want to talk a little bit about what you see as the positives and potentially the negatives of CRISPR.
So, first of all, let me take a step back and say, really, CRISPR and the whole technology around CRISPR-Cas9 is really a suite of technologies. And it's better to talk about the suite of technologies rather than individual instances of it. The suite of technologies allow us, to make it very simple, allow us to make edits, directed, intentional changes in genomes. We could do some of them with very cumbersome tools before. These new technologies have refined the capacity to do this. And just to go back to our example, again, remember the encyclopedia. Let's say I wanted to go to volume 72, page 314, and change ACTG, erase that and change it to ACGG. Of course, that could be highly relevant. It could be the difference between one human being and their brother who happens to have cystic fibrosis. That tiny change could revert or change a disease or the course of a disease. So these technologies allow us to become much, much more efficient at doing this. What the side effects are, whether you mistakenly change something else in another page is a question, although more and more we are realizing that they're quite specific, actually. So the question then becomes, what do we do? How do we use it? What are the limits of how we use it? So there are really three arenas that I think about in terms of their use. One of them I would call genetically modified organisms as broadly as you can imagine. So modifying, on one hand, crops, modifying mosquitoes, potentially creating new weapons against pests. So that's one arena, and that, of course, there's a huge debate swirling around that arena. What are the limits? How safe? Is there biosafety concerns there? The second arena is in humans, what we would call non-germline or somatic gene therapy. So you don't go into sperm and eggs. You use CRISPR or Cas9 to change the biology of stem cells, the genomes of stem cells, like blood stem cells, something I work on. So last week we used CRISPR. I'll give you one example. Last week we used the CRISPR system in cord blood stem cells. So these are stem cells from human cord blood. If you put them into another recipient, they would repopulate the entirety of their blood. So these are highly active stem cells in blood. And using CRISPR, we essentially changed the genomes of 90% of the cord blood cells. That number was inconceivable 5 years ago, to make a directed, deliberate change in 90% of cord blood stem cells. Inconceivable 5 years ago. And what's the relevance? Well, you could take a patient who has sickle cell anemia and give them a bone marrow transplant from their own cells and change the sickle cell gene. You could take a patient with thalassemia and so on and so forth. You could take a patient with immunodeficiencies and insert or remove or change genes that affect that and repopulate their blood with their own blood or cord blood where you'd made a deliberate change. So really a new universe. And then the third arena is obviously the most controversial. So not genetically modified organisms, not somatic gene therapy, but germline gene therapy. What if you introduced these kinds of changes into sperm and egg cells? And if you did that, of course, this would be heritable through multiple generations, potentially to every generation. That would be permanently changing the human genome, because once you've introduced that change, you can't take it back. Every time you reproduce, that would be. So the way to think about is that really to split it up into these three arenas and talk about the three arenas really separately. And that's, each one of them has a debate going on. But it really helps to clarify that there are really three applications or three arenas of application.
And your concern here?
Well, so if you just go one by one, if you make direct, deliberate changes in other organisms, the concerns are safety and biosphere. The concern is humility and understanding enough about the biosphere to have the humility to know what to do when you unleash powerful new genetic technologies in the biosphere. I am told, I don't know if this is for sure, I'm told that the mosquito has no use aside from being a human pest. But we don't know. We don't know. Maybe the mosquito occupies some niche in ecology that we don't know about. And the famous story of the butterfly effect story, we don't know and we won't know. There's a famous science fiction story of a man who goes backwards in time. It's a kind of a fantastical story. This was written 20 years ago. I read it as a child. He goes backwards in time, and the setup is that you go back in time to hunt dinosaurs. It's a sport, but you're told that you can only hunt a dinosaur which is destined to die within the next 10 minutes because you can predict all of this. So one of these, in this crazy story, this guy goes back in time, but the dinosaur he gets flabbergasted. He steps off the road and kills a butterfly in this story. And when he flashes forward back into his own time, the entire world has changed. This was the kind of conceit of the story. Everything that he knew about has changed. Nothing remains the same because he stepped off and accidentally nipped the wing off a butterfly or something. But the idea here is that. So that's the GMO piece. The somatic piece, the biggest concerns are danger to the person, to the recipient. What if, in trying to correct a sickle cell gene, you made the cells cancerous? You unleashed a mutation that was you didn't know about, and in fact, 5 years later, 10 years later, some kid that you had treated for sickle cell anemia using his CRISPR for bone marrow transplant all of a sudden came down with a lethal cancer. The concerns for the germline gene therapy are the deepest. Where do we draw the lines? Are we, what do we interfere on? Who is, we are now playing with the human genome in permanence. Is it to be restricted to diseases of extraordinary suffering? Are we going to allow ourselves to change, to remove genes that increase the risk of cancer? What about human traits like height? What about other traits? Are we going to start really playing with things that we were previously unavailable with?
How do we regulate this? Is this something the government's going to step into? I know we stopped this on a couple of occasions in the past. Is science going to march forward regardless of what we do?
Well, the government, a very surprising thing happened, to me very surprising. There was a national panel that met.
Shocking to me, actually.
Yeah, actually. And everyone should know about this. The national panel met, and I thought they would say absolutely not. So human germline genetic manipulations and manipulations on embryos, we should draw a strong line. But in fact, the panel met. It was composed very diverse. It was comprised of lots of thoughtful people, scientists, people from the legal world, patients, patient advocates, and they essentially said that in the United States it would be permissible to make germline changes under 3 circumstances. The circumstances were, and in fact, what's interesting about it is that if you think about it, they're very logical. They're the same that I described in the book, in the gene, independently. I mean, I had written the book 6 months before the panel had met. Number one is that it should involve extraordinary suffering. These were the exact words that were used, that there should be extraordinary amounts of suffering. This is not to be taken, in other words, this is not to be taken lightly. We are allowed to intervene where there's extraordinary suffering. I'll come back to that phrase in a second. The second one is that we should really have some sort of certainty that when we are making a genetic change, that we will indeed change the disease of extraordinary suffering. That this is not to be done with things that have light or weak links with diseases. Some genes have small, weak links. Some have very powerful links. And the third one is that it should be justifiable, that we should be able to justify it. There should be no other simple, reasonable choice. There should be no coercion. This should be free, justifiable, etc. But of course, if you think about it, all those three words, who defines what those words are? What is what's extraordinary suffering to you? What is justifiable to you? What is the exact strength of the link between the gene and the trait? So this is an arena that I was very surprised by. I thought there would be a bright line difference, and we would put a moratorium. But in fact, if the moratorium doesn't exist, we are now moving forward.
So obviously there's tremendous opportunities with the human genome, both in understanding and then potentially in therapy as we go forward. There are also some dark sides. And one of the things that I noticed that you dedicated your book to Carrie Buck.
Yep.
Tell us about the saga of Carrie Buck.
So that decision was made very early in the book, to dedicate the book to Carrie Buck. Carrie Buck, I found out, actually I didn't know, Carrie Buck was one of the first women to be sterilized by government mandate because she was thought to have genetic variant of imbecility. I'm using words from the 1920s and 1910s. In fact, when you look through the data, and now we know there was probably nothing of the sort, but she was stereotyped, and the claim made by the government was that she had a hereditary form of imbecility, and she was confined to a state colony where so-called imbeciles were held. And the fear, this was in the throes of the great eugenics movement, the fear was that imbeciles would breed and propagate more imbeciles. Carrie Buck, the case of Carrie Buck rose to the Supreme Court. And Oliver Wendell Holmes famously wrote that 3 generations of imbeciles is enough, and thereby forced her to be sterilized. She had one child who died. She would never have another child again. I wanted that to be the bright line reminder of where this could go, of our capacity to intervene on humans, on human heredity, where the dark history of that, and how quickly our world had devolved into that moment. I wanted to remind people, and therefore the dedication of the book.
Tell them about the evolution from that to run the rest of the world.
So interestingly, the idea of eugenics, the idea of using technologies, however crude, the idea of using technologies to improve the human condition through controlling heredity, was born in very exalted circles. It was considered a very, very good idea. Lots of people who had very benign intentions in the 1800s, 1890s, and 1900s signed on to this idea. We would call them, today, they would be among the great humanists, scientists, writers. And the word was coined by Galton, Francis Galton, who was Darwin's cousin. And Galton's idea was, this is one way to improve human race, the human condition, make us better by selective breeding. So this was born in England. Then it morphs. It comes into the United States, where it moves from selective breeding, intermarriage, to selective sterilization. And it's here that the idea of selective sterilization is given a kind of imprimatur of social acceptance, hence Carrie Buck, and hence tens of thousands of women who were sterilized and men who were sterilized because of epilepsy, because of so-called imbecility, because of a variety of things that turn out to be treatable, or in fact, shouldn't even have been defined as non-eugenic conditions. And then in its final form, it sort of moves again, and it metastasizes to Nazi Germany, where it moves from sterilization to extermination. So rather than sterilize people, exterminate them so that they don't have the capacity to produce more, and all in the name of that same word, eugenics. So that progression, I think, is very important to remember as we move forward. It's important to remember for me because my family has very much the marks, the fingerprints of the very things that were being exterminated. Had in some fantastical universe, those fingerprints, the fingerprints, the genetic fingerprints that inhabit and haunt my family in other places would have led them to their death or to sterilization. So it's important to remember these circumstances with a great deal, I think, of humility and thoughtfulness.
Let me change this tone of this for just a second. You've had a tremendous opportunity to read, study, meet interesting people along the way. If you had an opportunity to sit down and interview one of these people,
what a lovely question.
Who would you pick?
Tough
Darwin?
Tough choices. I think Darwin. The way Charles Darwin changed the way we think about the world is so fundamental that even to see him in his English cottage, I think would be a kind of a fantasy, a vision come true. The only person who competes for me is not his contemporary, but certainly a person who lived not far away from him, and is Dickens. So, Darwin, Dickens, you choose.
You're stuck in the D's.
I'm stuck in the D's, exactly.
So you have an incredibly talented wife.
I do have a wife who's more talented than I am, yes.
And she was a MacArthur Genius Award winner, a contemporary sculptress. How do your ideas bounce across each other?
It's a very, very interesting. So we rarely, I should say that if you're thinking that dinner conversations in the Mukherjee Z household are scintillating, they're not.
Well, you're in the
because
you're in the kitchen cooking, for God's sake.
No, it's because it's things like, did you remember to pay the bill? Did you? What about the car insurance? No, far from it. We, I think, have evolved a way of working together. She reads everything I ever publish. I try to see things that Sarah does. I'm in the studio. Her studio is not far away, and the kids come. They've grown up in this way. We critique the work, and we critique. She even reads my scientific papers.
Is she tough?
She's the toughest critic. Yeah. It's the old saying. It's better to be critiqued by your friends than by your enemies. You could extend that. It's better to be critiqued by your wife than your enemies. But maybe that doesn't hold. But I think we spend serious alone time with each other's work, and take it very, very seriously. And it's not the things. She will write margins full of notes on something I'm going to publish, again, ranging from scientific papers to something that might come out in the Times or in the New Yorker or come out as a book. When I go to the studio, I really, really spend time thinking about the nature of the work and trying to find out. The one place I think we meet is I like to ask, whenever I see something, I like to ask, what question is this something trying to answer? I like this way of thinking. It's helped me a lot. If I see a book, it could be a work of fiction. When I see a scientific paper, when I see a clinical study, the first thing I ask is, what question is this trying to answer? And is it answering it? Maybe partially. And the same thing goes for when I see Sarah's work. I always go to Sarah's work, and it's a very strange way of thinking about her work. I say, well, so okay, there's this massive sculpture, which could be the size of this room. Sometimes the works are the size of this room. And I say, the first thing I'm saying to myself is, what question is this piece trying to answer? And the same thing when I write something. What is it? What question is it trying? It's something that gives me a clarity about what I think.
I've got one more question, then we're going to throw it open to the audience. So get your questions ready. You recently wrote an article for the New Yorker.
Yep.
Essentially subtitled, "The Algorithm Will See You Now."
The algorithm will see you now. Yes.
Yes. And essentially, AI versus MD. Do you see that happening?
So that article generated more back and forth, controversy than virtually anything I've written, because I wanted to lay out some of the important questions that I think are coming. What are the important questions that are coming? The idea that we will use deep learning very broadly to change medicine is, to me, already is a moot point. I think we are already there. We will go there even more.
Absolutely.
And the resistance to it, particularly for certain kinds of knowledge, I think is futile. So the question really is, what is it that, the question it forces us to answer is, what is it that doctors do that deep learning systems cannot do? What is it that we do that's amazing and special? And there are many things. That's not one thing. One thing that strikes me is that we ask the question, why? Deep learning tools, machine, big data, interestingly, is still powerful at looking at patterns, finding, doing diagnostics, but it often it struggles with the why question. Why did this disease happen? Why is it that a particular trait is linked with a particular feature? Why does pathophysiology occur? So I think one of the things that deep learning, big data is really forcing us to do is reconsider what we're especially good at. Hypothesis building is something we're very good at. Hypothesis testing is what we're good at. Pattern recognition is we're actually okay at. In fact, it is our failure of pattern recognition. It is our failure. It is the chink or the flaw in our capacity, in our brain's capacity to recognize patterns, that allows us to be strange, creative thinkers. We look for exceptions. We look for things that you would otherwise bin in one basket, but we say to ourselves, well, wait a second, somehow that doesn't make sense. We're very good at that kind of stuff. So I think that's where sort of our medical integrated future with, as someone put it, our robot overlords will be. We will become accessories. We will become collaborators, but we will reserve for humans the things that humans do very well. Answering why, providing compassion, providing connection, contact, context, those kinds of things.
So just a parenthesis on that. This article was stimulated by a discussion that you had with one of our former speakers here, Sebastian Thrun, who came and talked to us and subsequently developed an opportunity to recognize the patterns for malignant melanoma.
That's right, yeah.
And which is becoming a clinical entity. And so that they now, with 95% accuracy, can diagnose a malignant melanoma from a benign lesion. So that was the impetus. And so the moral of the story is keep coming to these so you meet these people. Let's start over here.
Dr. Mukherjee, thank you so very much for your very stimulating and enlightening discussion today. It's great to hear a fellow Exonian who attended Modern College as well.
Oh, sorry.
I had a question related to just the power of the genome and our entering the sort of this new era. And I'm wondering what your most common kind of questions or misconceptions that you hear about genes and genomics, and perhaps the interface with the power that they have, and how machine learning and that approach can take to understanding what genomics is.
Well, I'll give you, it's not just a misconception. I mean, there are many, many trivial misconceptions, so I'll just neglect them for a second. I'll give you something that's actually really, I think, a concern for people who talk about precision medicine. So let me talk about that for a second, because the phrase precision medicine is something, again, I'm coming from the world of cancer, where the idea that you can use genetic information, so genetic precision medicine, genetic information to predict, to be able to treat with much more refinement and accuracy a patient, because their individual mutations will predict response. This idea was born in the world of cancer and now is spreading across other disciplines as well. But again, remember the example that struck me recently, that in the case of many human diseases, the genetic architecture of the disease is not just 1 or 2 or 3 or 4 genes, but tens of thousands of genes with small nudge effects. When you try to build a plausible biological model of these tens of thousands of nudge effects, we begin to struggle. So, for instance, again, to give you the example of height, I'm making this up, I'm speculating here. What if one of these tiny nudgers, responsible for 0.25 centimeters or 0.025 centimeters of variation in height, what if in fact that was a gene that led to the set point of your appetite? Right? So I'm making this up speculatively. The point here is that to make a plausible biological model, it's easy to make a biological model if you have simple connections between nodes, and you have a simple connection. You can say, okay, well, this gene affects the metabolism of your bone, makes bones grow longer, and therefore intervening on this will hopefully, not touch anything else, will spare everything else, will make your bones grow longer and become you'll get the height portion solved. But once you enter, one of the complications of real complex human traits is that there are tens of thousands of genes with small nudge effects and lots of something we call pleiotropy. And making a plausible biological model so that we can intervene on that might become tough. And if that becomes tough, then making medicines that work on these will become tough. So that is a challenge. I think this is one of the concerns and challenges as we try to make new kind of medicine occur, is that we might be bumping up against the genetic architecture of complex human diseases. And it's an incredibly complicated idea to explain to ourselves, let alone the lay public. But we have to do it because we've started using words like precision medicine. So I'm happy to talk more about it, but this is a tough idea and could be extended all the way. Alzheimer's disease, heart disease, things like this. Sorry.
Thank you, Dr. Mukherjee, for your amazing presentation. I want to thank Dr. Cosgrove for his great contribution to the field of medicine. And I know, Dr. Cosgrove, you're going to retire soon, and we're going to miss you. And I'm sure everybody here agrees that you'll be missed. So thank you for your service, and thank you for everything you've done.
You have a second life as an interviewer you could take over Charlie Rose's job.
I had to pay him a lot to do that, by the way, so thank you. He did it very well, just like we rehearsed.
My question for Siddharth is, Siddharth, I have two questions. One is a macro question and the other is a micro question.
Okay.
I've listened to many amazing people and read about them, including Satya Nadella of Microsoft, Sundar Pichai of Google, Ajay Banga of MasterCard, and Indra Nui from Coke, and now you, who have contributed amazing to the society. What is it about this beautiful country called United States of America that people come from India and many other parts of the world that allows these young, bright people to flourish and contribute amazing to the society? And I don't think that it's really something we can argue about. The fact is that they do. And my micro question is, if you don't, if you allow me, is if we study the genomics of these people, since that's your specialty. I know you are not a social scientist and you're not going to develop immigration policies or anything, but if we study the genomics of these people, which is your interest, what do you think we'll find? Because it might help us to bring even more smarter people to this country and make this country prosperous for next 200 years. Thank you for listening.
So the macro question I'll tackle. So I think this is, I'm going to borrow the answer, I'm going to borrow the answer from Sundar Pichai, which is Sundar was recently asked this question as well. Maybe we were together. Sundar was asked this question as well, and Sundar said, it's a little bit of a denominator problem. The denominator problem is that obviously you've chosen very kindly, you've chosen lots of numerators. People come to the United States because they want to, because they're driven by their dreams. People like me, they are driven by freedoms and perhaps some former freedoms that we used to enjoy. People are driven by the idea that there's a fundamental, or there was formerly a fundamental belief in the emancipatory process of science and technology in this country. And when we lose that belief, people will stop coming. I will stop coming. People like me will stop coming. So it's a very, very fragile ecology that was invented in this country 200 odd years ago, and that ecology attracted, like sunlight attracts plants, attracted people from the most adventurous people from the world over. We sought our adventure here because we thought this was the most adventurous place to be, because we couldn't imagine a universe. When I first came to college in the United States, I went at Stanford. I went to a professor's office hours. This was Paul Berg, who had won the Nobel Prize. His office hours, outside his office was a little sign-up sheet on a piece of paper, and you could sign up whether you were an undergraduate, a first-year medical student, whether you had won the Nobel Prize yourself. Just put your name down on that sheet of paper, and you could go and have a conversation with him about the future of genetics and whether recombinant DNA would change your particular field. That's the kind of adventure, the accessibility, the openness, and that's what made we're the beneficiary, we're the numerators of that process. So if you destroy the ecology, the sunlight will go away.
Thank you again for being here. I heard your conversation with Sam Harris on his podcast. That was also a very interesting conversation. And
two conversations, actually.
Yes, two. And you talked about how cancer is basically a genetic disease, and it's a problem of mutating genes, and that there are two basic ways that this can happen. One is just a simple replication errors, and the other are chemical that overtake, or toxins. And so I'm wondering what percentage, where do you sit on this controversy? What percentage do you think of each? And for the ones that we now think are just simple replication errors, how many of those might actually be caused by chemical or toxins that we just don't know about yet?
So it's a tough question to answer because there's not a lot of empirical data. I'll give you I'll give you one piece of important information here, and I'm going to go straight into controversial mode. The one piece of information here that's relevant is that, and I'll challenge anyone to, this is an interesting experiment. The statement that I want to make is that we have not discovered a single major chemical carcinogen, preventable chemical carcinogen, since the 1990s, 1970s. Not one. Okay? So you could say why. Why have we not? And the words are important. Single major preventable chemical carcinogen. We've discovered many chemical carcinogens since the 1990s. Their impact on broad human health is minor, each one of them minor. Nothing of the comparative effect as, for instance, smoking, right? So, and the question is why? The answer could be, number one, is because of a conspiracy, because we aren't being inclined to look in the right places. So that could be one answer. The second answer could be that it is death by a million cuts. So in fact, we'll never find major ones. The major ones have already been found, and that most of the impact is going to be in small effects, could be. And number three is that they don't exist. And that the rest of cancer, whatever the rest might be, and some people believe it's the major part, some people it's the minor part, the rest of cancer is going to be unfortunately the consequence of replication errors, errors in copying DNA, which are intrinsic to human cells. So the point that I wanted to make to Sam, and I want to make to everyone, is that we have to be honest with each other about answering this question. We cannot be wrapped up. We have to bring data to the question. We can't get wrapped up in individual proclivities in answering the question, because the question has enormous consequences for cancer. So I can only tell you right now that the evidence, at least to me, the evidence that random chance cellular replicative events cause cancer is quite high, and that we have not discovered major chemical carcinogens. So we're stuck in this moment where we're trying to find the ones that will solve the missing problem. You know, why are we getting, why we have cancer? We have cancer because we're aging as a population. But what are the carcinogens that are driving this? But we have to be honest in answering this question and dividing it up thoughtfully. That would be my, that's my general answer. Thank you.
Okay, over here, and then one more question, and then we're going to wrap it up.
So mental health, particularly in this country, has a huge stigma attached to it, and I wonder, as you looked at your own personal history, how that, your family history, how that impacted you personally. And then as we look at the genetics behind it, I wonder if you think that we may eventually be able to take mental illness from being looked at as a personal fault to being considered the disease process that it is, and then will we be able to force insurance companies to actually pay for the care people need?
Yeah, I think that's absolutely, those are very important points, and part of the reason to write the gene is to do exactly that, is to bring us up to a kind of understanding of mental health. I have very little doubt that that powerful determinants of mental health sit in individual genomes. This is not to diminish the role of the environment, absolutely not. We know, for instance, even with diseases where there's a powerful genetic link, such as schizophrenia, identical twins don't have 100% concordance of developing the disease. They may have high concordance, 10-fold, 50-fold higher than the general population, but it's not still 100%. So this is not to diminish the environment, but this is also to remind ourselves that powerful genetic links exist. It goes back to the same. So as far as insurance is concerned, this is obviously, or payment is concerned, this is a reminder that that we really need to understand and be empathetic to the way we are made up as human beings. The challenge lies in building that biologically plausible model. Because then, once we have a biological plausible model, that's where human creativity actually, this is what human beings are very good at. The challenge is there because with schizophrenia, just to give you one example, very much like height, very much like other complex human traits, we know that there's a powerful genetic link, and yet if we add up the individual contributions of all the genes that have been known, they're tiny. So this is the missing heritability problem in mental health, in schizophrenia in particular. We know that there are powerful genetic links, and yet we have not been able to find those genes that contribute to the heightened risk. And we have not been able to build a model that explains, a mental model, a biological model that explains why a child all of a sudden, has this terrifying, devastating breakdown in their mental processes. And if we can't build a model, we cannot find targets that are rational to be able to treat and to be able to tackle. And therein lies the peril and the promise of medicine applied to mental health. Building real models, building real deep understanding of how known genetic risk translates into disease. Thank you. Thank you for that comment.
Dr. Mukherjee, thanks for being here. In your discussion, you mentioned Charles Darwin's cousin. And from my reading, I know he also made contributions to statistics and also to meteorology.
Yep.
And people like Da Vinci also made contributions to multiple fields. And my sense is that today, academia has become far more specialized and far more siloed. And I'm curious if you think that's going to hinder our society's ability to answer some of the big questions that you and Dr. Cosgrove touched on during the discussion earlier.
Yeah, so very broadly speaking, I do, obviously I think that living in very, siloed academic enterprises can be a problem. That said, just remember that some people are, by definition, highly defined in their quests, highly defined in their goals, very defined in their ambitions. They don't want to. They don't define themselves as interdisciplinary. Others are profoundly interdisciplinary. They look for ways to connect fields. They're connectors. Malcolm Gladwell has a nice description of these two kind of phenotypes of humans. I think there's again, I go back to the idea of an ecosystem. I think a thriving intellectual ecosystem has both kinds of actors. You have the cardiothoracic surgeon who is astonishingly good at cardiothoracic surgery, but is a terrible general surgeon. But then you have to have the general surgeon who can move between surgical disciplines, surgical fields with a kind of facility that his or her colleagues might not have. And finally, you have the internist who sits and thinks about how AI is going to transform general surgery, who may not even have surgical hands himself or herself. So or a surgical brain himself or herself. So there's a way in which I think the diversity of human endeavors, and it's necessary to preserve the diversity of human endeavors. That if you lean too hard in one direction, you often inevitably lose the other.
Well, Sid, I want to thank you very much for being here today. You have enlightened us. You have challenged us. You have thrilled us with the potential for the future, and you've also added a note of caution. And I want to thank you very much on behalf of all. Please join me in thanking Sid. And just a couple of reminders. Our next speaker is going to be Ginny Rometty, who is the president and CEO of IBM, who's going to be here on November 16th. And following that, later at a date to be announced, John Doerr, who is the managing partner of Kleiner Perkins, a venture capital firm in California, will be here to discuss disruption. So thank you very much for being here. We look forward to seeing you in November.